Welcome to another edition of Health Innovators. I'm Paul Tunnah, and as you know, in this series, we've been spanning innovation in all kinds of different aspects of healthcare, from digital through to biotech. And today, very much focused on the biotech side. Real pleasure to have Eliot Forster here with me, who's Chief Exec of FStar Therapeutics. Eliot, thank you for joining me.
Absolute pleasure, Paul. It's always nice to catch up.
So you and I first met, I'm trying to think, many years ago now. And I think at the time you'd just come out of the big corporate career with Pfizer into the world of biotech. That's where I wanted to start. Take us back to kind of your journey through big pharma and how you've come into this role within biotech and how that's developed.
Yeah, so as you say, I'd spent 15 years coming out of academia and then into big pharma, GSK in the first instance, and then for a relatively short period, and then 11 years with Pfizer. And I was fortunate enough to be involved in some programs going to the clinic, and then moved into more senior management roles. And I guess all of that time, one gets exposed more and more to biotech, and it looks interesting. And it was a chance conversation, in all honesty, with an old friend of mine who was a prof at Harvard University, a guy called Clifford Wolf, who said that him and a few of his acquaintances were thinking about setting up a company, and wondered if I would like to be involved. And there was no specification of that, and indeed the company was just an idea. But I think it was that kind of seeded the thought that maybe the land of biotech could be of interest. And so one thing led to another, and sure enough, about a year after that conversation, I found myself as CEO of my startup company, Solace Pharmaceuticals, which was based in neuroscience, in particular looking for treatments for chronic pain.
Yeah.
It was quite a change, and I remember, if you'll bear with me for a second, the stark contrast between Monday, which, because it was an end of the month, happened to be the last day, and Tuesday, where I went from having a full secretariat, access to telephones, computers, IT support, HR, vast teams of people to work with to deliver drugs, to me in my kitchen with none of that. And it was a shock, I have to say. But what I'm delighted to say is that it was also an opportunity to learn a whole new array of skills and experiences, and I've never looked back. I would never say never about going back into big pharma or big biotech, but I think this is the place for me.
Yeah, and as you say, quite a system shock, I guess, initially when you move from the big corporate environment to biotech, and some challenges as you've outlined there. But kind of what did you find most exciting in those early days, having made that transition?
Yeah, so I guess it was both caused anxiety and exciting in equal measure. So it's the fact that you are the entire momentum of the business. When there's just one person, quite literally what you do is how the business moves, and there's no natural cadence. And of course, in big organizations, I'm sure it's not just true of pharma and biotech, but in big organizations, there is a natural momentum to those businesses that we all experience. When you go on holiday for a week, you come back and the business hasn't stopped, and so you've got a whole week's worth of work. If you go on holiday when it's just you, nothing happens. It literally just stands still. And so I guess in equal measure, that was really exciting, because you can have a direct impact on how the business is going, what you're doing with the science, and so on and so forth. And it's palpable. I mean, literally, you say, I'm going to do something, and it happens. But at the same time, that means that the things you decide to do really make a difference. And that's less the case. And I was pretty senior at Pfizer when I left. And just because of the scale of organizations, everyone's actions from the CEO down are buffered somewhat by the system. So I think that was it. The other piece is knowing that you don't know huge array of activity, and the classic for a kind of startup first-time CEO is when you get to talk about directors and officers insurance, and it's almost a guarantee that a first-time CEO he or she will come along and say, what about directors and officers? What is that type of thing? And I've had lots of conversations with first-time CEOs, and we talk about directors and officers insurance, and it's a kind of rite of passage, and you're in a biotech when you're having to figure that out for the first time.
Fantastic. And aside from that, was there anything that really surprised you, you were not expecting as part of that transition?
So one of the things that I'd always been interested in is the kind of progress of the programs, scientifically and ultimately clinically, medically, and I'd incorrectly assumed that that was going to be the single motivating characteristic for everyone involved, and of course it's not. And there are lots of different interests in the biotech world, and in particular because we have to have funding. When you come to the financiers, they are interested, of course, in the scientific and clinical progress of the programs, but because it's driving value. And I had to reorientate my thinking to that value driver alongside the scientific and clinical drivers. And that was quite a surprise. And clearly, 15 years on, I'm very used to that. But I remember at the beginning, it was quite a surprise. And there is a circular argument to say that if you deliver good clinical data, then value will follow, and so on. And it gets to be more nuanced. But it was the money stuff, I guess, that got to be interesting for me and surprising.
Yeah, and I guess that balance between that passion for science and patients and investment returns is there in the whole industry, but I guess it's a lot more acute in the biotech side sometimes.
Yeah, absolutely. And of course, in the fundraising part, which is a CEO's full-time occupation, actually, in biotech, even when you're not fundraising, you've got to be thinking about fundraising, as it were. And in that fundraising, it's really about translating the opportunity for patients in biotech into a value proposition, and a value proposition which is time-bound, not a value proposition that's 30 years hence. It's in the investment period, and that's a really interesting exercise, and it remains a challenge even today for me and the teams. It's finding the right translation of something which can often be highly sophisticated and involve science into something which will lead to value. Now, most of the investors that we interact with on a day-to-day basis are sophisticated enough to help with that translation, but they don't want to have to do it. They want you to do it for them, and then work with them in that respect.
Yeah, well, it's a very technical area, so I guess that's understandable. So speaking of technical, we'll come on to FStar in a moment and your science. I know you've got some really exciting stuff going on, but let's finish the story before we get there. So what's your journey been from those early days in Solis to FStar? And also I'm intrigued in how you've seen the broader biotech sector maybe change in that time.
Yeah, so we were both lucky and unlucky at Solis. So we were financed for the first time in 2007, and with 3.5 to 4 years worth of runway with that financing, so $19 million. And of course, just as we were closing that, the worldwide financial crisis was beginning to close in. Now, of course, because we had money in the bank, we were able to operate, but it had a direct impact on the next round. And unfortunately, our phase 2 program failed. So we were left with some preclinical assets. And in today's market, those would have then probably carried the company. But at that time, there wasn't sufficient liquidity around, and it was still pre the 2011 Jobs Act under the Obama administration, which really has caused the bull market in biotech ever since. And so what I learned was a lot of really tough lessons. So we eventually closed Solis down, and the process of that was absolutely horrendous at the time. But when I look back to it and the folks I was working with, we learned an enormous amount. And in fact, it almost should be a rite of passage for exec teams in biotech, and no one really wants to go through it. But it teaches so many lessons. One is about the reality of the connectivity between investment value and science, but the other is that actually you can get through it. No one dies at the end. you learn a lot and then can move on to the next. And so the next for me was moving to a European company. So Solus was based in Boston, a European company called Creabilis, which again was in the neuroscience and information space. And I was there for 5 years and really helped move that organization from a place where there was some good ideas to go into the clinic to be clinical stage and running clinical studies. I left, but just after I left, that was then sold to another inflammation dermatology company, which was listed and so on. So that went on from there. I left in order to switch therapeutic focus actually into a company called Immunocore, and I was there for three and a bit years. And again, and I sort of fall into this by accident, was there bringing something which was early, very early clinical and non-clinical into the clinic, and expanding the profile. And for each of these companies, of course, raising capital, and the difference between a preclinical or non-clinical activity and clinical, of course, in terms of capital requirements, is enormous. It's an order of magnitude different. And so for each of the businesses, raise capital in that respect. On leaving Immunocore, I was kind of debating as to whether I should do another CEO job, because I'd always promised myself, because they're 24/7, you're buried in it. I'd always promised myself I'd have a break and maybe become more plural. And a couple of things happened. Is some people who I very much respect said, "You must be nuts. You know, despite how you look, you're not old enough yet to move into a plural existence." And indeed, really, why don't you turn some of those experiences? And so I was fortunate enough to be contacted by a very old friend of mine, Jean-Francois Formula at Atlas Ventures, who said, "Look, we've got this company, FStar. It needs the transformation that you've done twice before, which is good ideas non-clinically into the clinic and build value." And so I took a close look at it, and in many ways, similarly to Immunocore, a bispecific in the immune oncology space, next generation thinking, and really got excited by the technology, by the folks who were there, and thought, look, there's an opportunity to do something here. And in particular, in the wider context that immune oncology had really evolved since the beginning of the last decade, to 2011 onwards, with the checkpoint inhibitors, and really the whole industry and patients are searching for the next generation. It's true that checkpoint inhibitors have worked brilliantly, and there are some patients who effectively get a cure because they're in disease control for decades, but that's a tiny proportion, and the vast majority, 70 to 80%, who often get forgotten about actually in discussion around checkpoint inhibitors, don't get that benefit. And so second generation approaches, which are wide ranging from cell therapies through bispecifics and into gene therapies, actually ultimately are really, I think, where we'll begin to cut heavily into that patient population who are not getting that long-term benefit.
Yeah. And I think just to reflect on that biotech journey, anybody that's made that entrepreneurial leap has made mistakes, has got their battle scars to show for it. And I think as you touched on there, that's enormously important, because having gone through that journey a number of times, I'm sure that's what they saw in you for FStar, was somebody with that experience in the biotech space. And you touched on there, obviously, that shift to immunotherapy and oncology. You touched on there the kind of science, but tell me a little bit more about the science involved in what you're doing at FStar and what's unique about it and how it works.
Yeah, so FStar is one of many companies that are dealing with bispecific antibodies for the treatment of advanced cancers. What I really liked about FStar is that the solution for the bispecific was so incredibly simple. In fact, when I first looked at it, it was so simple, I sort of was doubtful that it was real. And how simple it is the inventors, who were based out of Vienna in Austria, took a normal antibody, which has got what's called the constant region, so the bottom of the Y, if you imagine an antibody cartoon,
Yeah.
That constant or crystallizable region was always thought to be sacrosanct. You can't do anything with that because it will destabilize the antibody, etc., etc. And they were actually quite remarkable, and they said, well, actually, we think we can do things with it and not interfere with the antibody architecture. And so sure enough, they found that by substituting a few handfuls, so 15 or so amino acids in very specific locations in the FC and that bottom end of the antibody, you can create another pair of binders which have got the same binding ability as the naturally occurring binder. So I looked at this and I thought, hang on, we've got a few amino acid substitutions, nothing else. So the antibodies are basically a natural shape, yet you have this 2+2 tetravalent binders. And sure enough, that's exactly what FStar does. And so it benefits from the ability to make lots and lots and lots of versions of those, because actually substituting 15 amino acids, I'm not trying to belittle what goes on in the labs, of course, but it's substituting 15 amino acids. We're not bolting great other scaffolds on, or making other architectural changes to the overall antibody. It's basically an antibody. And so that works well. It also means they're easy to manufacture. We get to the clinic quickly, et cetera, et cetera. So all of that was intriguing. I'd also looked into the backgrounds of some of the folks who were in the organization, and also FStar itself. And FStar had a really successful background in taking this platform technology, so this 2+2 bispecific, and partnering it, and actually brought in at that time over $200 million in partnering income, and that's validating as well. And once you've looked at all of that, I thought, okay, here's something to do. Now, what wasn't the case was that FStar at the time, when I joined in 2018, didn't have any drugs of its own in the clinic. It was basically working on behalf of other people, and it was really that transformation that needed to occur to build we're back to the value question, to build that equity value for the shareholders.
Yeah. Which is interesting, the approach you outlined there, because people talk about disruption a lot, particularly in technology and digital. But what you've described is disruption really in the world of biotech. And I know from my university days how delicate protein folding and antibodies can be. And you can understand the perceived wisdom of, you can't touch that, you'll completely destroy the antibody. But being able to challenge that and say, well, why not? Let's try and do something different. That is kind of scientific disruption, isn't it?
Yeah, very much so. And even today, when people we talk to for the first time are looking at what we do, they just don't believe or grasp it, to be honest, because they're used to looking at pretty complex scaffolds in order to achieve even 1+1 binding, to be honest, but 2+2 is tricky. And then we explain, and we have to be careful to make sure they've understood, because they can still walk away with the disbelief, as it were, and think, oh, these guys must have added something to it, and genuinely haven't. And I'm full of admiration, because it's very easy, I think, to follow a line of common beliefs and stay with those and try and work around those common beliefs. But these guys, Florian Ruka in particular, was able to say, well, actually, I'm going to see if that belief system is wrong, and that's quite a thing. Fortunately, for an academic, he and his partner Gordana also were cognizant of the need to patent, and in fact, we have over 200 granted patents, and so we end up being the only company who can make any changes in that space to create new binders. And that's part of why we've been able to do a lot of partnering. And the really cool thing is it works in all sorts of settings. So one of our partners who are outstanding is a group called Denali Therapeutics, and they're based on the West Coast, working in CNS. And so we've worked with them, and they now are in mid-stage development in Hunter syndrome, again with this modified FC region type molecule, and they're protected and we're protected from intruders, with that simple change. So it's very exciting.
Well, I was going to ask you about the application because obviously immunotherapy, there's a lot of activity there. It's very broad in terms of application. So, in terms of your focus, I mean, it's interesting to hear that you're looking at stuff outside of oncology as well. But in terms of your focus within oncology, where is your initial focus at the moment, and what sort of indications?
Yeah, so one of the things about the architecture of the FC region is it's a more rigid and narrow structure, and it's actually perfect for the TCR, sorry, the TNF alpha superfamily, and how those receptors become activated is through clustering, and actually the FC modified FC region that we have, because of its architecture, really is ideal for clustering this superfamily of receptors. And so what we have done is actually stood on the shoulders of giants who've gone before it, and a lot of investment has gone before it in first generation, and looked to see which of those receptor families and classes we believe will be best suited to help that 80% of patients, as it were. And so we look at mechanism, we look at data that's coming from others. We obviously have an in-house expertise in immune oncology, and make some decisions about which pairs we want to bring together. And that's really only the beginning of the story. So let me take the example of our lead program. So our lead program, FS118, has got a PDL1, which is a classic, well-known checkpoint pathway, PD1, PDL1.
Yep.
We like the ligand because it's overexpressed on the cancer cells, and so we get some directing of our molecules to the cancer cells, which is great. And then on the other end, we've got LAG3. Now, first generation of LAG3 didn't work because people were inhibiting LAG3 on its own, and frankly, until very recently, those of us, and there aren't that many companies who are working in the LAG3 space, were considered, to be idiots, because what are you working in that space for? It doesn't work. Well, actually, what we knew internally, and what others who've continued to work know, is that the first generation told us that you have to do either a combination or better, a bispecific against the classic checkpoint pathways plus LAG3. And indeed, on Sunday, just on the 6th, BMS published phase 3 data,
Yep.
significant phase 3 data with a combination of Nivo and their LAG3 antagonist, and showed a doubling of progression-free survival significantly in melanoma patients. Yep. And now the whole industry has said, oh, LAG3, that's exactly the right place to go. And we stuck with our guns from the beginning with that, and because of the data we'd seen internally, we really believed in that LAG3 pathway, and we also believe, of course, that a bispecific approach to that is going to be important. So it's as nuanced as that. There are some other things as well, which, if you'll bear with me for just a second. So lots of people instinctively, and I was like this, half a decade ago, will believe that if you've got two binders, what you just need to do is get, as high a binding affinity, so as sticky as possible for each of those binders, and bring them together, and then you'll have this, tight cross-linking. You bring two cells together, they'll be really sticky at each end. Unfortunately, biology is just not that simple, and it turns out that if you do that, if you make them both as high as possible in some settings, then actually you begin to self-defeat. Right. And so we have another program, FS222, which is a PDL1, again, the antagonist that I discussed again, and against an agonist target called CD137 or 41BB. And it turns out that if you tune those both very high, then you begin to self-compete in the setting of the tumor. And so you get a peculiar bell-shaped dose response curve. Now, some people's technology, that's all they can do, because that's the control they have over the platform. We're very lucky, again, we're only substituting 50 amino acids, so we can have a look at a wide spectrum. And it turns out that what we have in 222 is a really, really high affinity PDL1. So it sticks to PDL1 and stays, but actually as a mono binder, are pretty poor. The sort of thing that the head of research would send the guys back to the labs, it needs to be better binder to CD137. But when you bring them together, the avidity, so the overall binding, is absolutely off the scale, and you don't get that bell-shaped dose response, so you don't get that binding. So it's I know that's another layer of detail, but we're really fortunate in that we're also able to control and examine avidity versus affinity. And in the land of bispecifics, that's the key that unlocks the door.
Yeah, no, it's an interesting story, and it shows there's still, as you might expect, a strong element of serendipity and challenging perceived wisdom in all of this science, because we're still learning how it works.
Absolutely, yeah, and we all are guilty of having, or at least I shouldn't say we all. That's hideously unfair. I am guilty of having relatively simplistic models in my head as to how all of these things are interplaying between different cell types and different receptor types and so on and so forth. And of course, that model may be 100% wrong, and even if it's right, it's only going to be partially right, and the environment is much more complex. And of course, it's also dynamic. So one of the things we showed with the most advanced program, FS118, was that not only do we get the pharmacology, so we get a cross-linking and we get actually a shedding of LAG3, which is really rather cool. So you're not just blocking, but permanently altering that inhibition pathway. But it's also interesting to see then the dynamic of how that's impacting the actual immune cells. And we see an uptick in immune cell activity and expansion of them. And of course, disease control, which is what we're looking for. But of course, it's not as simple as the straight pharmacology on one receptor. There's then pharmacology on two receptors, how those receptors interplay. There's then what does that do to the actual immune response, which then in turn will be impacting on what it is we want to do. So the dynamics of that system are extraordinary. I cut my teeth. My first job in industry back in Glaxo in the very early 90s was in PK/PD modeling.
Yep.
And I remember we were building with a chemical at the time, a so-called Emax model, so an S-shaped model. And myself and a guy called Jonathan Palmer had taken some data from ondansetron, which is a nausea and vomiting drug, a 5-HT3 antagonist. And we had a room which was the size of most people's offices, full of computers. And we set the thing off on Thursday afternoon, knowing the data would come out with this model, which I could do on my phone now, on Tuesday. And so we got this, tiny simplistic thing, and it was a one-to-one relationship, single compartment models. Really, really simple. And we've gone from that to these dynamic systems where, we're playing with the immune system and trying to figure all of that out. And what we need to do is continue to learn through that as well, and also think about it in a systems biology way, not just a straight pharmacological way. And everyone does. I mean, this is not me breaking any news to anyone here, but it just says that even in what is 30 years for me, but even in that relatively short period, we're learning how little we know, I think, to your point.
No, it's really interesting, isn't it? You get a lot of, as you know, really good science in preclinical, but when you stick it into clinical, you stick it into the human body, which is very complex. You just don't really know what's going to happen. Yeah, it's very hard to predict.
Exactly. And all of the the patients are of course different. In immune oncology, of course, there are different diseases. Each of those diseases has a different stage. And what's really interesting is the way in which the patients are treated impacts the nature of the disease and how one can treat that in the next step. So again, if in the advanced program we have FS118, which is in phase 2, we have a patient group there who have previously been treated with a checkpoint inhibitor, had a response, and then become refractory, and they have a specific biology compared to those who haven't been through that process. And we're treating that biology that's emerged as a result of a prior treatment. And so it's, again, it's all good because we're, everyone's working on it, and we'll figure it out. But it is complex. There's no doubt.
I think people forget that it's a moving target. So an individual's cancer will evolve over time, change over time, and that's what you're trying to deal with. We'll come back to, I'd love to hear a little bit more about your hopes and aspirations for the impact on patients. Before we talk about that, though, I'm just intrigued to hear, for what you can share, about how you see the growth of the company, because it feels like the biotech model has maybe changed a little bit. And maybe 15 years ago, if you went into phase 2 alone, you were ambitious. If you thought about phase 3 alone, you were completely insane.
Yeah.
Whereas now it feels like biotech is starting to behave a bit more like big pharma, looking at acquisitions or different types of partnerships. You had the combination with Springbank Pharmaceuticals, for example, last year. So what can you share about that future growth trajectory and how you see partnership or potentially acquisition coming into that?
Yeah, so I think we need to step back to the kind of macro environment first, Paul. So what's different from when I entered biotech land in the 2006, 2007, and now is that we've had a decade of basically a bull biotech market, and accelerated even further, frankly, because of COVID and the importance of biotechnology and pharma in tackling the pandemic. And so there's a lot of capital available, there are a lot of sophisticated investors, and more of both of those available now. And so biotechs can think in slightly different ways than we did when we first started, which was get some clinical data and exit the company, as it were. And of course, NASDAQ in particular has become a great landing spot to get to sophisticated capital and lots of it. And so for our journey, we've had to do quite a lot since 2018. One is to find a portfolio of our own, and I'm delighted to say from our platform, we've now got 3 programs in the clinic, which is really exciting, and the team has done a blinding piece of work over 3 years. But we also needed to get to NASDAQ, and I guess one of those things you learn, and it's easy in hindsight, is our board at FStar. We talked in December of 2019 and said, look, we're about to go into a presidential election year. We didn't know about the pandemic, of course, at that time. And if you don't get out, the doctrine was, if you don't get out onto the market by March, maybe April or May, then forget it, because everything's going to close down while there's an election, et cetera, et cetera. So we essentially decided to prep for an IPO on NASDAQ, but at the same time recognize it was likely to be this year that we would do it. On top of that, we got to JP Morgan, and there's a little bit of coughing starts, and it's, wow, what is this thing coming over the horizon? Then, of course, we all know by February we knew we were in pandemic trouble. So I think we and many others assumed that would be it, markets closed, et cetera, et cetera. And of course, we couldn't have been more wrong, because of course last year was a unbelievable year, as has this year continued to be.
Yeah, it's accelerated
for IPOs and activity in the NASDAQ. And so we got really active, and there are basically for a biotech there are 3 pathways. One is the classic IPO, the other is to go these days through the SPAC method, which is effect a reversal, an RTO, or there's an actual reversal, which is the method we ultimately ended up with, and whichever one gets there first should be the one probably to pick. And we were really fortunate to come across Springbank Pharmaceuticals and their team, who were really super, and we've still got some of those guys on board, and we work with them. And what was interesting about them is obviously they had a NASDAQ listing, but they also had a phase 1 next generation oncology product. Now, obviously not from our platform, but it's a STING agonist, which we're really excited about, SB11285, and we'll report data on that in the next couple of months. And so we talked to them, and virtually like this, we merged the 2 organizations, and that gave FStar access to the capital markets on NASDAQ, and we traded for the first time on the 23rd of November of last year, and that was really exciting. And of course, it's taken us to the next stage. So by the end of last year, we then had 4 programs in the clinic. We were NASDAQ listed, and so that journey of transformation that we'd embarked upon, from the end of 2018 was going well. And I think everyone probably knows we also then closed the financing a couple of weeks ago, and of around $75 million, which carries us through into 2023. And we also continue with our partnerships, which are excellent. So, the company itself, 4 programs listed, has really now poised to make that difference to patients and then add that value to shareholders. I think one of the things we talked about a little while ago was, is that a pathway to do more than once? And I think there is certainly the capital available. for organizations to combine forces. It's not something that's on the horizon for us, but what we can do, because of where we are and the amount of capital available, is actually to say, well, actually there is potential to take, you know, 1 or 2 of our programs ourselves all the way through to market. And as you say, you know, a decade ago, that was, you would be considered a crazy to think about that. And more companies do it. You know, my old company, Immunocore, is doing that, you know, with GP100, their most advanced program. That's really exciting. You know, and I'm aware of others who are thinking in exactly the same way. And so for us, obviously, it'll be led by data, but the patient population I mentioned a little while ago, which are those patients who become refractory to checkpoint, they have no other options. So they're going to palliation or some chemotherapeutic regimens, which obviously we all know are far from ideal. And so if and when we get, you know, data in the middle of next year from that patient set, and we get a benefit that we hope to see, then that would give us a very fast track to market. I mean, that's why we're in that particular space. It's deliberate, and you know, a few handfuls of patients will be enough to get an approval. And so we would need to be thinking, you know, by the end of next year, we'll be thinking, okay, well, we're not now that far away, and because of NASDAQ and because of the amount of capital that's available, all of those things are feasible. Of course, what tends to happen in many cases, however, is at that point, you know, one of the very big fish comes along and says, oh, we quite like the idea of that, and then you end up in a kind of defense position. But you know, what we do and what we are doing is focusing on our own pathway for now, of course.
And are you seeking big pharma partnership for any assets at the moment then?
No, not right now. I mean, we do talk to the big pharma fairly regularly. There remains a lot of interest in the platform. I want to be careful with that, because I don't, you know, 4 programs for an 80-person organization is already quite a heavy lift. And so we don't like to distract ourselves too much. But certainly, if there's not a route through a fast track, small numbers of patients, then as those clinical data emerge, I think that will certainly be something we'll want to look seriously at, because you then need that firepower and scale to be able to drive through. We got some very interesting data in non-small cell lung cancer, and as you know, that's an enormous market, but it's also very competitive, and one needs to, you know, look hard at how you deal with that. So all of that's in the pot, as it were.
So let's just look to the future then. It's to round off the interview, and first of all, in terms of the potential impact for patients, I'm interested to hear about, I guess, your aspirations, your hopes. And I know it's tricky because you can't predict what data is going to show, and you've got to be careful to manage expectations. But certainly in areas like immunotherapy, you start to hear people daring to use the word curative therapy, and how far could we go with this? What are your hopes? Where do you think this could take us?
Yeah, so let me just talk about the kind of macro environment first. I mean, I think because of some of the reasons we discussed earlier, you know, there's never going to be a cure for cancer, because cancer is not a single disease. It's a multiple disease with different origins, viral, non-viral, et cetera, et cetera. However, I do believe that, and we're already in this land now, that one can turn an advanced cancer into a chronic disease, in which, through careful management of regimens, what a patient can have a life expectancy which is not dissimilar to one who doesn't have cancer. And we're getting there now. Of course, one of the things that everyone tends to forget in drug development is you've got to think about safety and tolerability as well. And there's and we need drugs that can be given for a long time and be well tolerated, because ultimately for a patient, it's not just about controlling their disease, it's also about quality of life. And that's why, for example, with the advanced program, if we could substitute in a reasonably well tolerated drug, or a well tolerated drug instead of chemotherapy, then that's obviously going to be a physician and patient and their family's choice. So I think on a macro level, we will see great advances. I'm really excited about the power of a wider understanding of the genome, and then genetic manipulation tools. So gene therapies, cellular therapies, in a background or alongside the bispecifics and next generation treatments that we're all developing. And I think those combination approaches ultimately will give many more patients that long-term control of the disease. I think in order to get a cure, we need to go back to the beginning and begin to understand, what it is, where are the mutations coming from, what are the origins of those, what's it doing, and we know some of it. I mean, if HPV is driving, increase in head and neck cancer. Head and neck cancer has emerged now as the 6th most prevalent cancer in the US. And that's largely on the back of virus. There are other environmental things. So I think it's a life course which we'll get to in order to get to the cure, as it were. But I'm very optimistic about that, and the smart people and the way in which investors are thinking now is all about that. So that's exciting.
Yeah. And then for you personally, with your 10-year plus journey now in the field of biotech, and you think about the next few years, and obviously beyond the patient impact and the excitement that brings, what kind of motivates you? What are you hoping to achieve the next few years?
Yeah, well, clearly, I mean, my central mission is to continue to try and help FStar on its journey to make a difference to the patients that we're interested in, and we haven't had time in this interview to go into the details of the needs we're aiming to address, but it's suffice to say that we collectively and I continue to be committed to make that difference to patients, and I think the value will follow that. And I think clearly that's front and center for me, and will continue for that journey for as long as needed. I mean, if I look out beyond that, if there is a beyond that, I think the thing that I really get excited about is obviously the new technologies which are beginning to emerge, and the new pathways that are beginning to emerge. There are companies like Nucleome, Enara, Cellmabs, to name a few, who are just like 3rd generation thinking now, which is really exciting. But what's even cooler about all of those are the people who are involved in them, and I think ultimately, if I can get to a point where I can help first-time execs, first-time CEOs not make the mistakes I've made, and not trip themselves up in the same way, then I think if I can get to that point and add some benefits, then that really will be the thing that for me will be most exciting. I think in parallel, I'm on a personal level, I'm very interested in ensuring that we take this opportunity as a society post-pandemic to come back in a different sort of way. And if you might for just a second, I was born in Sunderland. By dint of profession, I've ended up living in a different parts of the world, and now different parts of the UK. And one of the grim features of that is my life expectancy is greater than my family who stayed in the northeast of England, and that's true of Liverpool and Manchester, and actually some parts of the north coast of Kent, who are similarly deprived. And there's a kind of economic and health outcome relationship. And I've recently taken on helping an organization in Liverpool that is aiming to make a difference in that setting, make a difference to patient outcomes and the economy simultaneously. And for me, that journey is really important. And if I can bring any of my business experiences to help that, and I know that's not a 2 or 3 year campaign, that's that's a generation, then I think that will be time very well spent as well.
Yeah. Well, that's something I would definitely support, because I also grew up in the northeast. For me, it was York before I came down south for university. So yeah, it's an important topic, and I agree, it's something that we'd like to see some movement on the next few years. Eliot, it's been, as always, it's been great catching up with you. I'm glad you decided to stay in biotech. I think biotech needs you for at least a few more years, and wish you continued success with the various different programs and the data that's coming through. Thank you very much for taking the time.
It's a pleasure. Thanks, Paul. It's been lovely to speak with you.